Hi, I'm Esther Comellas and I'm a Marie Curie postdoc fellow at Northeastern University. In this video, I'll give an overview of our modeling of the influence of limb flexion on cartilage growth. Joints are essential for movement, and their correct formation is critical for a healthy life. Alterations in the mechanical signal or in the transduction of that signal in chondrocytes is responsible for many developmental joint malformations. Yet, the exact role of motion or loading on joint formation is not fully understood. So, how do mechanics affect joint formation? To answer this question, we propose a combined experimental and computational approach. Axolotls regrow limbs throughout life, and these are morphologically similar to human limbs. Understanding how biomechanical forces drive joint formation in a regenerating axolotl limb holds promise for the study and treatment of joint deformities in humans. Predictive computational models allow exploring certain scenarios that cannot be easily interrogated via experiments. They can provide useful insights into the mechanobiological mechanisms driving joint morphogenesis. This study focuses on how we can start to predict the influence of movement-induced mechanical stimuli on humerus shape through finite element modeling. In our finite element model of growth at tissue level, the bone rudiment tissue is modeled as a biphasic poroelastic material, which consists in a fluid-saturated porous solid. Growth of the solid part is given by the sum of a biological contribution and a mechanical one. We assume that adaptation to mechanical loading increases with cell number. Then, the biological growth is proportional to experimentally measured chondrocyte density along the proximal-distal axis of the humerus. Experimental studies have shown that dynamic compressive loading promotes cartilage matrix production. Therefore, we define mechanical growth proportional to the compressive fluid pressure induced by the loading on the surface of the humerus. The starting point in our simulations is an idealization of the humerus geometry at the moment of joint formation. We apply dynamic loading to simulate the flexion-extension motion on the developing joint due to the action of the limb muscles, and then look at the predicted shape due to tissue growth. We examine predicted growth for normal joint flexion extension and for growth without mechanical stimuli. We observe the effect of incorporating mechanical stimuli in our computational predictions of the final humerus morphology. The growth increment shown here is a non-dimensional measure of the tissue's volumetric growth. When there's no mechanical stimuli, the predicted humerus shape is proportional to the experimentally measured chondrocyte density. Our model captures the effect of impaired mechanosensing on tissue growth, roughly matching our experimental observation of axolotl limbs. Notice the shape of the humerus. We have predicted the little bumps observed in the healthy control case that are not present when we block mechanics in the experiments. Note that there's two ways of doing this, through denervation or blocking cell mechanosensitivity. Uneven tissue growth for the normal limb flexion is driven by the non-homogeneous fluid pressure map created in the humerus tissue due to loading. What's interesting about this is that, due to the nature of the poroelastic material behavior, only compressive dynamic loading produces such results. Let's look at this in a bit more detail. Static loading generates an initial pressure distribution that quickly dissipates as fluid seeps out of the bone rudiment. Dynamic loading produces a pressure map with fluctuating but sustained pressures. This is what is driving growth in the normal limb flexion extension case. Even with all the simplifications and idealizations, our computational model is able to capture for the first time the effect that the type of loading imposed has on tissue growth. This is due to the incorporation of the fluid flow component introduced by the poroelastic formulation. These results are an exciting first step towards the development of a more detailed computational model that will be able to predict the effect of mechanical stimuli on the distribution of biochemical growth regulators driving tissue growth. To learn about how we plan on quantifying the effects of such biochemical growth regulators in our experiments, check out my other talk on the multiscale analysis of macromolecule synthesis. We want to link mechanical loading to biochemical stimuli, and these to tissue growth in a single model of limb development. Why? Because it will provide a powerful predictive tool that, alongside experiments, will help us better understand the biophysical mechanisms of joint morphogenesis.